a little bit about my motivation for getting into using deep learning to study RNA. And so this RNA language models that we've worked on to predict mutations in the ribosome, which I'll tell you about, to improve how the ribosome works. So mutations that improve RNA function. So again, because I think it's important to try to understand the motivation for this, I'll tell you a little bit about where this project started. So we are interested in how the ribosome can synthesize proteins encoded by messenger RNAs. And so this makes these sequence-defined polymers that the ribosome can make. And that the ribosome uses this chemistry of amide or peptide bond formation. And it has two sites for this, where there are these adapter molecules called tRNA. There's an A site and the P site. You'll hear those two mentioned a lot. And they use this acyl transfer chemistry to make to have this growing polymer chain. And in proteins, there are 20 plus or minus a couple of additional ones out there, but 20 natural L-amino-alpha amino acids that look like this. And the R group stands for many different types of chemical groups from this 20. And when these are made into these polymers, protein polymers, we can get really interesting proteins, ones that carry oxygen, that can fluoresce, all sorts of different functions in biology. Let's see if I can get my arrow to work here. So if we think about proteins as a chemist, as a sequence-defined polymer, it's defined by the messenger RNA code, and we can get these very interesting proteins out. We're, as chemists, very interested in changing what kinds of these R groups or side chains can be incorporated into proteins. There's the 20 natural ones, and starting with work from Peter Schultz about 15 years ago or so, we now, or more actually, it's now possible to change this to dozens, if not hundreds, of different groups. So we can get a lot of different interesting chemistries going on in proteins using this. And what we're interested in doing is not changing the side part of this chemical group, the side chain. We're interested in changing the backbone. And so this is how, if I go back a slide, that's how the polymer chain is put together, is along the backbone. And we're interested in changing that chemistry. And the reason to do this is that we can get new materials properties in these polymers. We could have them be useful for therapeutics, smart materials, a lot of different potential applications. And these are just some examples of types of what we would call monomers that we'd want to put into the polymers: beta amino acids, cyclic betas, other types of chemistries. The challenge is right now, the ability of us, our ability to use the ribosome to incorporate these is very limited. It's really just short polymers or peptides. It's very dependent on the sequence context. Getting many of these in a polymer is very difficult. And so what we're doing is we're part of a National Science Foundation center. It's called the Center for Genetically Encoded Materials, and the website is in the lower left here. And our goal is to try to make the ribosomal system better for incorporating these non-natural backbone monomers. So my role in this is a mini lab collaboration, and we're interested in the ribosome, which I'm showing over here on the left. And the ribosome is composed of what's called a small subunit in blue, a large subunit that's kind of here in purple and gray. And these come together with these molecules called transfer RNAs or tRNAs to polymerize the protein on the large subunit using the mRNA on the small subunit. And in order to get this to work, we have to think about three things. I've talked a little bit about the chemistry, but in order for the chemistry to work, we want this new chemistry to be going on in living cells. And so we need to have this system be orthogonal to the ribosomes that work in cells. So that's what I mean by orthogonality. And then, as you'll see as we go through the talk, we need the ribosomes to be stable in order to support this chemistry. So, And again, since I can't see all of you when I'm doing it in the presentation mode very well, feel free to jump in if you have questions along the way. Okay, so this sounds kind of like an engineering project, but one of the things that's always true about biology is we still don't understand the biology as well as we need to. So as we try to engineer the ribosome, we can discover new fundamental aspects of the biology of protein synthesis or translation along the way. So protein synthesis and translation are terms that are for the same thing in my talk here. So I'm going to give you an example of how we want to change the chemistry, and that's the possibility of incorporating these types of chemical monomers called aminobenzoic acids. And I'm showing these here. So as opposed to normal amino acids in proteins, these have this kind of ring in the middle, this aromatic ring. And by having this ring in there, it gives these monomers, when they're in polymers, different properties. They can fold into interesting three-dimensional structures. And they can also have properties like this aramid here, which is fire retardant. So lab coats that you might have in the lab, if you want to avoid having, your clothes catch on fire, you might wear a lab coat that has this kind of polymer in it. And so we're kind of interested in these materials properties. The problem is right now the ribosome doesn't really polymerize these monomers very well. And so these are reactions in a test tube that were done by the Suga group, and they were comparing to methionine or MET, that's a normal amino acid, and even getting a single one of these into a polymer really decreased the yield. And they really had to work on the conditions to even get this good of an incorporation. So there's a problem getting these in, and we wanted to try to understand why this is a problem. And so we wanted to see how these monomers or substrates would bind in the catalytic center of the ribosome, something called the peptidyl-transferase center, or PTC. So I want to take a little step aside here to tell you, how do we look at this nowadays? So nowadays we use, instead of X-ray crystallography, we're using an approach called cryo-electron microscopy, or cryo-EM. And there's a Nobel Prize in this very recently, because we're now able to use this approach of using an electron imaging, the electron beam, to get very high resolution structures of machines like the ribosome. So this is at a resolution of 2 angstroms. And for comparison, when we think about length scales, a chemical bond in an organic polymer is, carbon-carbon bond is like 1.5 angstroms. So we're getting pretty high resolution here. And what this allows us to do, it used to be that cryo-electron microscopy was called blobology, sort of blobby maps, but now we're really at high resolution. And so to give you an example of the kind of details we can see, so what we're able to do is generate these maps of effectively electron density. They're electron potential maps, but we call them electron density because we're crystallographers at heart. You can see all these details for where the RNA is. So these are nucleotides, protein side chains. You can see water molecules. You can even see ions in these maps.
Professor.
So very detailed. Yeah, does somebody have a question?
Yes, I'm asking just to get context. One would wonder, we're using the cryo-electron microscopy and getting this level of resolution, 2 angstroms. This the intention is getting the structural sort of representation of the ribosome, or we haven't, or we are talking about the RNA.
Yes, so the ribosome is actually made of and the bacteria is made of 3 different RNAs of different sizes and 50 proteins, more than 50 proteins. So it has about 250,000 atoms in it amongst those 3 RNAs and those over 50 proteins. And so what I'm showing here is just a zoom in. So if I go back, here, let me see if I can go back here. this is just the map, but this is kind of the whole ribosome, an image of the whole ribosome that we, 3 dimensions that we can get from cryo-electron microscopy. And within this image, we have 16S RNA over here with a little over 20 proteins, 5S and 23S RNA over here with over 30 proteins. And we can get all of this information about where these roughly 250,000 atoms are by using cryo-electron microscopy. And this is really important. We're using it to really try to understand the chemistry of protein synthesis. So
Okay.
that's where this is coming from. So these RNA nucleotides are part of the ribosomal RNA in the ribosome. And this is just how these particular nucleotides are positioned in the ribosome. Does that help?
Great. Yeah, very helpful.
Yeah, and so with the cryo-EM, we can now, because of this very high resolution, at least in the best parts of these maps, they are experimental and some parts are better than others. We can distinguish things that chemists are interested in, like hydrogen bonds versus van der Waals interactions. We can get better defined stereochemistry. We can infer, various kinds of interactions with ions and water and things like that. So this is very useful from a chemistry point of view. So getting back to these aminobenzoic acid monomers that don't go into polymers very well when they're used by the ribosome, we were able to use cryo-EM to get structures with these substrates bound in the catalytic center. That's what these are the parts that we're most interested in, and they're kind of right in this region here, which is where we have the highest resolution parts of our cryo-EM maps. And so this is in angstroms, and so you can see that the exterior is maybe not quite as high resolution, but right in the part we're interested in, it can get to very high resolution. So what we see when we look at this is just to show that we have reasonable resolution for these monomers docked into the catalytic center, such that we can see, atoms, kind of where the atoms are positioned. And that helps us to understand the chemistry better. So the first thing we learned, so the way that these tRNAs have to bind, or the substrates bind in the ribosome, these substrates, is they're attached to tRNAs, and the tRNAs, being RNA, they like to form base pairs, and the end of the tRNA actually forms a base pair with the ribosomal RNA. So this is actually part of the ribosome, and this is part of the substrate that comes into the ribosome. And this base pair forms, this is a natural amino acid. We can see that even though these aminobenzoic acids don't go in very well, they do form this base pair. This is kind of a necessary thing to happen. They also, another part of the ribosomal RNA forms a pocket, kind of a chemical pocket, where the side chain part of the monomer packs in there. And you can see that also happens with the aminobenzoic acid. So these are fine. So it doesn't seem like this is the problem. And again, we're able to use cryo-EM to get this very detailed kind of analysis. So where are the problems? The problem is that when we zoom in on where the actual chemistry is taking place, the aminobenzoic acid monomers can't get close enough to do this nucleophilic, so what's called a nucleophilic attack, this nitrogen attacking this carbon to form a new covalent bond. So this is roughly what you would see with normal amino acids. These are 2-methionines. But the aminobenzoic acids don't get close enough. So they're kind of not able to do this chemistry because of that. Yeah, so the orientation is just not favorable. So another thing that has to go on, and I wanted to go through this because it gives a little bit of a flavor of the complexity of RNA. I'm showing you a lot of static images, but like all biomolecules, things are moving around. And for peptide bond formation to occur, some of the nucleotides in the ribosome have to rearrange. And so I'm showing 2 here in this kind of hot pink color that when there's, before the reaction can happen, they're kind of blocking or protecting the substrate in the P site. And when the A site part comes in, these nucleotides move out of the way, and then this monomer rotates so that you can actually form the chemical bond. And this just kind of shows that rotation and the movement of this uridine 2585 out of the way. And so this kind of gives you a sense of the flexibility of RNA in some parts. And so then when we looked at these aminobenzoic acid monomers, what we see is that they don't move out of the way. So they don't have this, what's called an induced fit. They're not able to do that. And so what happens is that means that this nucleophile here, or the nucleophile is not able to get access to where it needs to make a chemical bond. So that's another problem. That's really the kind of the first problem that we noticed, is that this induced fit is not allowed by this aminobenzoic acid. A third thing that happens is there's a lot of water involved in this chemical reaction. I'm not going to go through the details here, the chemistry, but just to say, by cryo-EM, we can see the positioning of these waters. This is with natural amino acid substrates. We see all three of these waters, 1, 2, and 3, that have been proposed to be involved in this chemistry. If we look at aminobenzoic acid monomers, we can only see maybe one of these waters. And so the other two just are not there. And just for the chemists, we think that maybe just the pKa of these monomers allows any reaction to happen at all, because they're not positioned very well. That was a lot of kind of chemical detail. I wanted to start there because this just gives you a sense of the motivation for where I'm headed with the language model. And I won't go through all the details here, but just basically the aminobenzoic acid monomers don't fit in the ribosome very well. And we are thinking about ways to make better monomers that we could use with the ribosome as it is. But what if we could make mutations in the ribosome that would allow these to fit better? This is where we're headed with these language models. So I want to give you an example of what happens if you do make mutations in the ribosome. This is in the ribosomal RNA that we're going to change. And this is to be able to incorporate things like this beta amino acid. So here, again, the detail is you have an extra carbon atom here, and that makes it very much harder for the ribosome to incorporate. And so Alana Shephard's group a while ago made a number of mutations in the ribosomal RNA, that's the sequences down here, to change how the substrates, so this is the A site and P site, the ends of those tRNAs poking down into the catalytic center, how those might be able to incorporate this monomer. And so they made 12 changes in the peptidyltransferase center, which is a lot. This is a very large RNA, it's 2,900 nucleotides, but when you ever, so almost 3,000 nucleotides, but when you work in a catalytic center of an enzyme or an RNA, this is a lot of mutations to make. So what ended up happening is, even though it worked a little bit in cells, when we purified these ribosomes, what we found is that much of this catalytic center was disordered, or just we couldn't see it. It was not very well formed. And so this is, again, a cryo-EM map in gray. And any of the model that's sticking out of the gray means that it's not very well positioned, and it's not very well ordered. And so this was kind of a damaged ribosome in a way. And if you look at ribosomal RNA, what I'm showing over here is called a secondary structure. It kind of looks like an airport terminal with little things coming off. This is how the RNA looks when it makes base pairs, and then this folds up into 3 dimensions to fit inside the large subunit. And it's this region here, near where the peptidyltransferase center is, that's not very well assembled here in this P7A7 ribosome. So the lesson we learned here, and then this is just kind of details of some of the things that might be disrupted. Again, this is an RNA, 3-dimensional RNA structure. And what RNA likes to do in a lot of cases is it likes to stack bases and also form interesting hydrogen bond networks. And so these are base triples instead of base pairs. You have a third base coming in, and these would be disrupted based on the mutations that they made. And that may be one reason why this doesn't fold very well. This is the natural wild type sequence that I'm showing here. So but what's the lesson? The lesson is that if we want to do things like mutate the ribosome or use approaches like directed evolution, this is where you can make like libraries or just combinatorial libraries of mutations and then select for those that give you a new function, like catalyzing new reactions. We have to not only think about the biochemistry or the activity that we're interested in, but we also have to think about the structure of the ribosome and make sure it assembles well. Let me just take a quick break here. Does anybody have any questions right now before I go on? Or I know this is very getting into the chemistry, but this is what got us going on these language models. So I thought I'd share that. So so we need to select for proper assembly or folding of the ribosome in addition to function. All right, so I want to come back to this little triangle I had. I've been talking, we want to have new chemistry. And so what we realized is we also have to think about how stable we make the ribosome. So this gets back to this process that Frances Arnold won a Nobel Prize for this work, where she developed ways of using directed evolution of proteins. And what this means is, again, you can take a protein, let's say you have an enzyme, and you want to have it do a new chemical reaction. Well, what she realized was that it was important often to start with a protein that what we call thermostable, that actually works at very high temperatures, say 60, 70, 80 degrees Celsius. And what will happen when you do this and you start making mutations that give you this you're trying to get this new activity, often it will change the stability of the protein, decrease it. And so you want to start with a thermostable protein because let's say you started with one that works at 80 Celsius. After doing all the mutations, it might work fine at 37 degrees or body temperature. And so that's why people often will start with a thermostable, what I call a scaffold or a protein that's thermostable. And so we wanted to know if we could take a similar approach with the ribosome. So we want to make an engineered ribosome. So maybe if we stabilize it first, we can do a better job of engineering. So the challenge is we can't simply substitute the RNA part, the 23S RNA part, from a hyperthermophilic, say, bacterium, a bacterium that maybe grows at 80 degrees Celsius. We can't do that. Can't just put that into, say, E. coli. This is the bacterium that we're often using in synthetic biology, and this grows at 37 degrees. And so what we need to do is we can't do that because the ribosome has 50 components and it has a very complicated assembly pathway. And so the hyperthermophilic RNA won't assemble with the rest of that in E. coli. And so that's the first challenge. We need to basically work with the E. coli ribosome to make it more stable. And the second challenge is the core of the ribosome, where this catalytic activity goes on, is the peptidyltransferase center is nearly universally conserved. So what does that mean? That means in every form of life on the planet, the nucleotides that surround the area where this chemistry goes on are almost always the same. They almost never vary. And so that makes it hard to decide, well, what can I change if I don't have any examples that ever change? So we started by using what's called a phylogenetics, again, using sequence information. That's, there are a lot of groups out there sequencing environmental samples to see what kind of microorganisms are there, bacteria, archaea. And those sequences can be used to, can be aligned with each other. And this is a database where you can do that. It's called the Silva database and has high quality ribosomal RNA sequences. You can look this one up and use that to try to find places where we can make mutations. So again, coming back to this P7A7 ribosome that was to put in the beta amino acids, and this part is kind of disrupted, we were interested, we're kind of lucky in a way, because that part that was disrupted is the last part of the ribosome to fold. So when you take this 23S RNA, those are in the kind of the rainbow colors here. This is kind of the order in which it folds up into 3 dimensions. It first forms this kind of secondary structure parts, and then it will fold up into 3 dimensions. And so we can focus on this red part here, the peptidyltransferase center, to look for mutations there. And so we found a couple, and that's actually interestingly where one of the tRNA substrates binds. So again, the tRNAs are these L-shaped molecules that bring in the monomers that we want to polymerize in the ribosome into the A site. And there's this 2 nucleotides right next to where the tRNA binds that we found some variability when we looked in the alignments. So this is a schematic of those that secondary structure. Again, we have base pair. This is called a stem loop, and we have base pairs followed by kind of a loop structure. And the way this is encoded based on these alignments is that if it's black, it's not conserved. This could be an AU, or it could be changed. If it's red, it means this pair is always there. And then if it's in all organisms have these particular nucleotides. And then if you look in blue, these are almost always in every organism, except for about 5% of archaea have some changes there. And so what are these changes? Well, they change U's to C's. And what's interesting about these patterns, when you look at these phylogenetic trees, is that they're in these what are called thermoproteota. These are archaea, these microorganisms that grow at very hot temperatures, up to boiling temperature of water. It's amazing that they can grow at that temperature. And so the changes from U's to C's is kind of enriched in these organisms that grow at very high temperatures. And so, maybe we could change the E. coli sequence to put those C's in instead of the U's. So we did that. And I'm going to tell you how we tested whether this worked. So this is our biochemical experiment where we purify the mutant large subunits. Again, remember the ribosome has the small and large subunits that come together. That's kind of shown here. And then we make a very short peptide that binds to another part of a protein called luciferase or nanoluciferase. And if we can make this peptide, then we can make a luciferase that is catalytically active, and we can add a small molecule, and this will give off light that we can measure. So this is kind of how we test whether these subunits are active. And what we can do with these, because they're purified, is we can heat them up, cool them back down, and then test them in this in vitro or test tube translation reaction that has everything else needed for translation. And what we see with wild type, if we take the purified 50S, heat it first to 60 degrees, cool it back down to 37, or these may be even done at 30 degrees Celsius, you lose a lot of activity. But if you now substitute those U's for a C's, you can recover that activity. So that really showed that we're able to get more thermostability by making, actually I'm going to go back, by changing those U's to C's in the E. coli sequence. So changing 2 nucleotides in like 2,900 nucleotides gives us that effect. We can also look at these subunits in a more of a physical way. So these are experiments where you basically have different densities of sucrose solution in a tube, and you use an ultracentrifuge to separate out particles. And long story short, if you have a very sharp looking peak, you have a very well folded particle. And as you can see, after heat treatment, they start to spread out a little bit, and it shows that they're damaged. But what you can see is that that's the red line for the wild type case or the natural sequence. But putting in these mutations, you can see that the heat treatment, they don't fall apart quite as dramatically. So this is just evidence that we've improved the thermostability of the ribosome. So that's great. But what about this P7 ribosome? We wanted to make these mutations so that we could maybe rescue the activity of mutations like in this case. Unfortunately, that mutation was not enough. And so we still were not able to rescue this. And so this made us think more about this question we had about directed evolution and making a more stable ribosome. We kind of ran into a dead end with phylogenetics. We couldn't find anything more just looking at sequences by eye that we would want to change. So we wanted to know what other tools we could use. And so this is where now we get into the deep learning part. So I know that was a lot of kind of motivational parts there at the beginning, but I wanted to just give you a sense of why we got interested in deep learning. And this is where we got interested. So if you think about deep learning, this is a figure from the ESM fold paper, a protein folding paper. I don't know how many of you have kind of seen this part. This is a language model. So it has a protein language model, or I put large language model, but it's really a protein language model. And then on top of it is built a whole kind of network, a neural network that is very similar to AlphaFold, the other program that was developed by DeepMind for predicting protein structure. But in the AlphaFold uses those kind of sequence alignments I mentioned before, whereas this is just a language model. And so by analogy, you can think of this as kind of like the GPT kind of model on one end, and then ChatGPT built on top of that. So ChatGPT, it's not a language model itself, but it has all sorts of extra layers on top. And so by analogy, ESM fold is kind of like ChatGPT in the sense that it has a language model as a foundation, and then it builds on top of it, and in this case uses known structures of proteins to help train the model. So we wanted to know if we could at least make an RNA language model instead of a protein language model. So there's a catch, and this is a paper that came out in Nucleic Acids Research. If you use the NCBI, the PubMed, you can find this reference. And it's a great paper. When will RNA get its AlphaFold moment? So AlphaFold is great for predicting three-dimensional protein structures, but RNA is much harder because, and this is what was kind of laid out in this paper, the RNA databases are just really, they're woefully inadequate to the task. They have very few high resolution three-dimensional structures. The sequence, again, I mentioned like microorganism databases and things like that. The sequences are not very well organized. So it's very hard if you want to think on a computer science point of view, the data you feed into these models needs to be very well curated and organized. And that's just has not been the case for RNA. So there are 3 databases that are out there that people have used. Silva, there's one called RFAM. It's kind of analogous to PFAM for proteins, but it's not as honestly as well organized. And then another one called RNA Central. And so the conclusion of this paper, if you read it, is that we're not going to have a model for RNA folding this decade. So, okay, so there's a big challenge for the computer scientists out there. So what we decided to do is try to tackle at least parts of these problems so that we could at least try to make a language model. And so the first thing we did is we decided to use what's called the Genome Taxonomy Database. And this is a worldwide effort to try to take genomes from microbes and organize them into a phylogenetic tree that's kind of as rigorous as we can do. And what's helpful about that is that when you think about microbial sequencing, some microbes are a lot more popular than others or show up a lot more in sequences like E. coli or, maybe other enterobacteria in our gut. And so we don't want to have that overrepresented. And so the way the taxonomy database deals with that is they have, let's say there's 20,000 E. coli genomes, but it'll only pick one as a reference genome. And so it helps simplify analysis by having reference genomes instead of looking at all the genomes. And so we use this to build a new RNA database. And so we took search tools from RFAM searched the Genome Taxonomy Database. And so what we could do is generate much more highly diverse RNA sequences, or a database of RNA sequences. This is a particular kind of RNA called a riboswitch. This is a TPP riboswitch. You can see that we get, several fold more sequences with different nucleotide similarity or identity than you can get using the next state-of-the-art database. And even for something as commonly sequenced as 23S ribosomal RNA, we do pretty well in terms of getting new sequences, especially for the microbes called archaea. And so there's imagine you got to asking me to give a talk based on this Yekaterina Shulgina or Kate Shulgina et al. Paper on bioRxiv. And this was a key part of how we were able to make our language model. We built a new database. So this database is really important for us because not only does it give us more...
Professor Jimmy,
Yeah.
there's a question on the previous slide.
Sure. Let me get back here.
Yeah, yes. One wonders, since we have RFM existing as a global database, but we want to come up with the GTD, I am asking myself, what is the contribution of RFM to the creation of genome as we want to put up these sequences together?
Yeah, so if you go look in RFAM, one of the things that it does that's very helpful, it's a community-wide annotated database. And what for any known RNA that somebody's interested in that has a three-dimensional fold, you can, or even sequence information, you can generate what's called a covariation model and or matrix. And so basically what this does is allows you to search sequences for other RNAs that have a similar pattern. So RFAM has thousands of different covariance models for different types of RNA, but they don't actually have that many examples of each of the RNAs, right? So that they have these covariance models that you can use to then go search for additional types of those RNAs.
Oh,
okay. So this is a bioinformatics kind of tool that uses hidden Markov models and things like that to look for what look like similar sequences. Does that help?
Sure, yeah, makes sense.
Yeah, and so you can go to you can maybe even in the methods of our paper, you can kind of see what we did in order to find these, and also go to the RFAM site itself. So it's a useful database from that point of view, but the problem is it's not very rigorous in terms of being systematic. And so that's where the Genome Taxonomy Database can help us, is to be a bit more systematic about where we find these sequences. And so the other thing about having the GTDB is that we can start to assign phenotypes to those RNA sequences. And the one we were interested in was optimal growth temperatures of the organisms. So all those genomes come from organisms, and we were able to take a different machine learning model that predicts the optimal growth temperature of a microbe. And we're able to, so in this database, only about 15% of the organisms have temperatures where we know that they grow. But since this model came out in 2019, there were some more organisms, and what we could show is that those new organisms could be predicted by this machine learning model. Their growth temperature could be predicted very well. And so that gives us the ability to do the following. We could make a new database that we call Gtdb Acquired RNa with environmental temperatures. So we could assign growth temperatures to all the organisms we're interested in.
Does the connection of phenotypes to RNAs happen at the database level? We are not yet at the level of deep learning.
Yeah, so they happen at the level of the GTDB, the Genome Taxonomy Database, has genomes, And each of those genomes, we can assign a growth temperature for the organism that genome encodes. And so then, because we have the growth temperature for the genome, we then have an optimal temperature for the RNAs encoded by the genome. That's how we got the temperatures. Does that make sense?
Yes, yes.
So I'm just going to show the archaea here as an example. What you can see is that with the around the edges are sequences that we got from these various organisms, and it's on a log scale. But if you just think about the temperature aspect, the databases that are available, it's pretty scattered in terms of what information is there. But using this machine learning model, we can now have predicted temperatures all the way around the archaea, including identifying what could be like new thermophilic organisms that hadn't been appreciated before, or like over here, for example. And so this is a kind of a very useful database from that perspective. And again, because
Prof, there is a question here in the chat.
Yeah.
How stable are the ribosomes with all the chemistry going on due to the different microorganisms and their mutations?
Yes. So our assumption is that the ribosomes coming out of an organism that's predicted to grow at, say, 80 degrees Celsius, that ribosome would function at 80 degrees. But I think the questioner has another interesting aspect. The ribosomal RNA is not just 4 nucleotides, the A, C, G, and U. Sometimes they're chemically modified by the organism, and that those chemical modifications can improve the function of the RNA, maybe help them work at higher temperatures. Unfortunately, our approach does not allow us to look at those modifications. We can only interpret from the genome sequences the A, C, G, U nucleotides. Does that help answer the?
I hope maybe if he has a follow-up question, he will put it up.
Sure. Good.
Yeah.
So we now have genomes with associated optimal growth temperatures, and we can extract RNA sequences from those genomes and assign growth or optimal temperatures to those RNAs.
Yeah, Professor.
Yeah.
Just a follow-up question, Gabriel here.
Yeah.
Yeah. Well, you of course you talked about when the mutations are, for example, at about 80 degrees Celsius, we expect the same response. But what happens if it doesn't, meet in those lines? Either it falls short of the degree and the other partakes of the higher degree. What happens to the RNA in that process?
So if I'm trying to think of, let's see. So for our purposes, so we're inferring the optimal growth temperature based on either experimentally determined growth temperature for the organism or the growth temperature that we're getting from our other machine learning model that was published by another group. And so we're assuming that ribosomes will function optimally at the temperature that the organism grows at optimally. But we don't actually have to be, as you will see, when the way we use these temperatures, we didn't have to have a super high precision in those temperatures, because basically what we did is we bend organisms based on whether we use the whole database or organisms that were predicted to grow above 60 degrees Celsius. And so if we're off a little bit in predicting a temperature, it doesn't affect our machine learning model or the deep learning model that I'm going to tell you about that much. So hold that question and see if it makes sense when I get further along. Okay. So we built the RNA database. And so now, okay, how are we going to encode RNA for the language model? So again, we really focused on this language model part. And when you look at the protein language model, the amino acids have to be converted into what are called tokens for the language model. And so basically ESM2, if you look under the hood, it has tokens for all the amino acids plus a few extra things there. And it also includes a few special tokens in its language model. But basically, all these tokens they're kind of just lookups for high dimensional vectors that would represent that amino acid. So a few hundred dimensions, for example, to represent a leucine, or in a different vector to represent alanine. And the idea is that these language models will orient these high dimensional vectors based on similarities to each other, and patterns will be kind of embedded into that very high dimensional vector space. So proteins, this is how they were encoded. What do you do with RNA? So RNA, we think, is going to be very different. And the reason RNA is going to be different is that its folding properties are different. So this is from a biophysics paper showing that when you kind of zoom in on RNA, and you can, if you see where my cursor is, you can see that they like to stack on each other. The bases, when you look at nucleotides, there's the kind of the backbone part and then the base part. The bases like to stack on each other, and the bases are what we have, the letters representing the names part. And so biophysicists have shown that by simply knowing what is stacked on what, they can predict the thermodynamic stability of an RNA. And this is just an example of a case where they made some improvements to a model, but it's really the fact that, like, this UA pair, how it contributes to the stability of an RNA helix is based on what's above it and what's below it. So it's based on that GU pair above it and the CG base pair below it. And so when you think about the UA pair, you also have to think about the neighboring pairs. And so, and this is like the dominant aspect of RNA folding is base stacking. So if you don't understand base stacking interactions, you cannot predict RNA structure and function. So this is just another figure just to show how well using this nearest neighbor model works for predicting RNA stability secondary structure. And just a couple different figures from that paper. I mean, the correlations are quite good. The R-squared values are quite good. And so another aspect of this, thinking about base stacking, or just thinking about the names of nucleotides, you also have to think about they can form different kinds of base pairs. So I'm showing the standard Watson-Crick pairs here that you would have in an RNA helix, the GC pair or an AU pair. They could be flipped, so you could have CG and UA. And then there's this other type that RNA likes to make that's called a GU wobble pair. And this is a picture from this book that it's very hard to find, but it's a great, Wolfram Sanger, Principles of Nucleic Acid Structure. It's my favorite figure from this book. And one of the things you can see when you do it is there are other ways of making base pairs with the same nucleotides. So these are 3 other examples of AU pairs that are not Watson-Crick. Here's another GU, here's another GC. So when you just look at a sequence, you can't always tell exactly what kind of pair it's going to be in, even if it looks like it's positioned across from what would form a Watson-Crick pair. So this is just a complication where a language model could potentially give a better result, just because you're looking at just the linear primary sequence of the RNA. So what we decided to do when we think about the encoding, I'm going to go back. And so when you take a protein sequence, you basically encode it into a token library like this, and then these are assigned vectors. What we decided to do with RNA is we decided not to do, this is like the 5' end of the RNA prime end. We decided not to use the same kind of encoding that was used for proteins, which would be like a single nucleotide gets a token. And what we decided to do, I'm going to skip paired, we decided to use triple nucleotide encoding, where, for example, this A is encoded based on its neighbors as well, right? That green line is for that A, whereas this A would be different because it has 2 different flanking nucleotides. And that's kind of based on the idea here. This A has has a very specific context. So maybe I'll have to pick something different. Maybe I'll pick this. This may not be the best example to show it, but for any nucleotide, it has neighbors that are going to change how it's seen. So like this G, for example, this G has two U's surrounding it. That G has a C and an A. So those G's are not equivalent, and they would not be equivalent in the encoding that we're using for our language model. Does that make sense to everybody? So this is very different than proteins. And so it's coded as a sort of a triple nucleotide, and then you step one nucleotide at a time. And so the advantage of this is that each nucleotide, for example, that G, is represented 16 different ways in our vocabulary, right, in the token library, because there are 4 different nucleotides on the one side times 4 different nucleotides on the other. So that gives you 16 different ways to encode each nucleotide. And then at the end, you have, a few more tokens that you have to deal with when you're at the end of a sequence. So that's how we encoded RNA for our language model, really based on RNA biophysics and our understanding of RNA biophysics. And if there's anything that you remember from this talk, this is the one thing I want you to think about, is using what we know about physics and RNA, but the way I used what we know about biology or physics to build your models. And in this case, it's really understanding that base stacking is critical for RNA structure and function. Okay, so
Professor,
yeah,
about not RNA, but I think the protein encoding that happened when we had the tokens.
Yeah.
In language models, we tokenize, and my thinking was about, after tokenization, we create perhaps the vector embeddings, and sometimes we think about the positioning of the words, and we have the position embeddings. And I'm curious, do we need to care about the positions of these, where these nucleotides are coming from at the level of encoding? Do we need to think about positioning? And also
Yes.
from
So just like with any language model, we also include, if we're using a transformer network, we're also including positional embedding. So the positional embedding is on top of this kind of embedding.
Okay.
Right. With the transformer, we used a transformer network. And so we converted the nucleotides into this vocabulary, that kind of embedding. But then on top of that, we actually used a rotary embedding, which you probably have heard of or may have heard of, on top of that. So this is the innovative part of our model. Everything else is very much like what other language models do, like the protein or even the English or whatever. The architecture is very similar, and I'm gonna show you what we ended up doing. So before I do that, as you can tell, I have gray hair. And so when I learned how to program, how many of you can guess which language I learned how to program in first? What would be your guess?
I would say C or Fortran.
Fortran. So I learned how to program in Fortran.
Oh.
And so getting into, and I've had to pick up things for cryo-EM along the way, but I was still the most comfortable in Fortran. So coming into making language models and deep neural networks and transformers, of course, that code is not in Fortran. So so just hold that thought for a sec. So we figured out how to encode the RNA. It's really RNA biophysics. But then to develop an RNA language model, you of course come back to papers like this and train a model to predict the next token using transformers. So this is the attention is all you need paper, which is of course very famous. But this, of course, is not written in Fortran, this code. And so yeah, I could do a few things in Python, but not very well. So I ended up going to a YouTube series by Andrej Karpathy.
Yeah, sure.
And I don't know if you've ever watched any of his
Yeah,
videos, but
yeah,
he is
building
great.
and GPT. Mm.
He's great. And so I learned how to do the coding by watching Andrej Karpathy videos. And he also put on GitHub this Nano GPT, which is a very stripped down, very compact version of the GPT language model. And that was extremely helpful for my coding. So this is the second major thing. So RNA biophysics, Andrej Karpathy. So this is what really got us going on the language model. And so basically what I could do is take his Nano GPT code and tokenize RNA using the triplet encoding and get a very functional RNA language model out. And so basically what we got is with this triplet encoding, I took the Nano GPT code and I figured out through trial and error, again, because I'm new to coding, how to incorporate things like this rotary positional embedding. And this gave us very functional RNA language model. And so then, because I know we're coming closer to the end, basically what we could then do with these language models is we have about 800,000 sequences in the GARNET database. We could train a language model using these sequences. And this is like, just think of this as like all sequences. And then we could take that pre-trained model and fine-tune it on RNA sequences from thermophiles, really from GARNET hyperthermophiles. Again, these red ones from here. And then after that, we would sample complete 23S ribosomal RNA sequences by just querying with the beginning of the 23S ribosomal RNA. And I want to just give a couple of examples. They're in the preprint, but just to give you a sense of what this means. So we're querying with sequence over here. And then the model has to be able to predict or output all of this sequence that goes around all the way to there. So we're inputting about 384 tokens or nucleotides. I think, let's say nucleotides, and it has to predict another 2,600, roughly 2,500 nucleotides. So it's pretty good. The model is able to generate this entire sequence. And so we have the normal model trained on all sequences in the hyperthermophile, and then we can compare those two models. And we don't just generate one sequence at a time, because you worry about things like hallucination or whatever the word is you might want to use. So we generate sets of 1,000 sequences. And by generating a set of 1,000 sequences, we can then do statistics on the generated sequences. And if we run into artifacts, hallucination, by generating 1,000 models, we can identify whether we're doing that a lot or not. And then what we found is that we could get the generation process working well and use the sets of 1,000 sequences to identify mutations to make. So that's another key thing that we did, is we didn't just generate one sequence at a time. We tried to generate sets of 1,000 to be able to do statistics. So I want to give you very quickly, one of the mutations that the model output that we should consider was this U2477. And we mutated that to a C. Again, we used the same assay I told you about before, the in vitro biochemical experiment, and we could show that the 2477C mutation it was fine with ribosomes that we purified from cells, but if we did a 65-degree pre-incubation, it retained more activity than the wild type. So it does provide thermostability. And for comparison, these are the two that we found from phylogenetics. We don't see any additional stability by adding this on top, but that could be a limitation of our experiment here. But the key point is that we are able to identify, using a deep learning model, a mutation that would make the E. coli ribosome more stable. And if we just look at sequences by eye, these are what are called web logos. And this is bits. The higher the letter, the more conserved it is. And if you kind of look at these sequences, the ones that we were interested in, it's really this one here. Yeah, it's enriched a little bit, but by eye, I wouldn't necessarily pick this one out to mutate. So the model did something more than I would have done by eye. And this just gives you a three-dimensional structure of the piece of the ribosome to show what this nucleotide is doing. It's actually contacting a ribosomal protein, which is kind of interesting. So this is one example of how we use the model. I'll give you two more examples just to show that. So we predicted a number of other mutations in this region that when we made them, we could also get a more thermostable ribosome. But when there were mutations here that were predicted, they didn't actually make the ribosome more stable. Now, I think the reason here is that you notice that 4 out of the 5 of these are involved in base pairs. And our thinking about, and the reason we didn't do this one to begin with is that RNA secondary structure forms first, and it's the tertiary structure or the more three-dimensional part of the structure that's less stable in an RNA. And so our thinking is that changing just standard Watson-Crick base pairs, those are already very well stabilized in the E. coli ribosome. And so we only had really one mutation in this that was potentially of interest, and it didn't work in this case. But in the other two cases, it worked, it worked great for 2477 and the Helix 68 case. And when you think about the combinatorial, we've got 2,900 nucleotides to work with. To be able to get a success rate like this, where we have to biochemically validate our results, was very helpful to us. And so what we think of this RNA language model approach as really a hypothesis generator. And then we go back and we test the predicted kind of mutations that we want to make experimentally. But what the language model does is it makes the number of experiments we have to do much, much, much less. So now I'm going to zoom out and tell you where we're headed next. Again, we're still very interested in making these non-natural backbones. And kind of the big picture, what gets me very excited is if you think about life on our planet, only 3 polymers have been subjected to kind of evolution, or organisms have done this. One is proteins, the other is RNA, and DNA has also been done. I'm not showing an example of DNA. This is another RNA. And if we can get the ribosome to make these new types of polymers and they're encoded by messenger RNA, we could potentially evolve entirely new kinds of polymers for technology. And that's kind of the end of my talk. I've talked about work from people in my lab. The earlier work was Zoe Watson and Fred Ward, who are now gone off to do other things. Kate Shulgina and Connor Langberg and Amos all contributed to the language model project. This was actually done as a hackathon within the Innovative Genomics Institute. And the CGEM project is a collaboration with these labs. And we've also worked with Jill Banfield and her group. And so with that, I will take any more questions you might have. And I will stop sharing.
Thank you very much, Prof. This has been very interesting and exciting. For me, my first question would be about its context and clarification about the takeaway that I'm walking away with, is that the language model built was to identify mutations within these ribosomes such that we can make more stable ribosomes. There's mutations that are more stable.
Yes.
Okay. Then another thing I would ask about myself, maybe this was looked into or maybe it wasn't. What can be said about attention? Like visualizing it, if there is some sort of visualizing the attention, is there something that can be said about it? Because we know it's a powerful movement of the transformer, and maybe
Yeah.
Yeah.
So we haven't kind of looked into the attention maps with the RNA in depth